This is Cannaramic, and we're bringing you cannabis like you've never seen it before.
Today, we are with the esteemed Dr. Alison Muitri, and he is going to be talking with us about cannabis, CBD specifically in autism. And we're really delighted and excited to have the opportunity to talk with you today, Dr. Muitri.
Thanks, Felicia. It's my pleasure to be here and share a little bit of what we do with your audience.
Awesome, awesome. Well, it's very rare that regular everyday people get to hear from an actual person in the lab, like doing that work that we all get intimidated when we see you do it. And I actually told you that I became a science geek. I discovered my inner science geek talking with you, so I hope everybody else does too. Tell us, first of all, a little bit about your background. How did you get to researching CBD and autism?
All right, so it's a long story, but I'll make it short and precise.
Okay.
So I am a geneticist by training. I got my PhD back in Brazil, and then moved to US for my postdoctoral training and become a professor here at UC San Diego about 10 years ago. And my lab was always interested on studying neurological conditions such as autism. And we are lacking a good model for these conditions because these are human specific. Most of the biomedical research kind of focus on animal research. And so for a while, we were lacking this model because, I mean, things that happens in autism, such as lack of eye contact or language or socialization, is totally different in any other animal. So my lab turned into stem cells. We use human stem cells as a model to recreate the autistic brain in a dish. And this is a technology that was developed by a Japanese researcher called Shinya Yamanaka. He showed us that it's possible to reprogram a cell from your body into this embryonic stage. That's what we call cellular reprogramming. And I can take any cell type from your skin, from your dental pulp, from your eye, bring it to the lab, reprogram them, and then they will become stem cells. Those stem cells can now be guided to differentiate or to specialize in different cell types or tissues in the body. And obviously, my lab has been focusing on the brain. So we guide these cells to become brain cells.
Okay, now stop there because there's a lot that people need to unpack. So, first of all, you mentioned that autism research is typically in animal models.
Right.
So that's not the same as human beings. So when we're looking at all the information that we're learning about autism, a lot of it is not actually based upon actual human being interactions, correct?
That's correct. I mean, especially the research that requires experimental biology. So this is when you need, for example, to stimulate a specific neuron or to create a genetic model. We can't do that in humans. So we're relying in animal models. And I think my message here is that those animals models are not perfect. They have limitations. Humans communicate through language. Animals communicate by sniffing each other.
Right.
So it's different.
It's different. So with regard to the stem cells, what you're saying is your lab can take a sample of cells from my arm, take it into your lab, make it like reborn again, like a baby cell again, without any definition as to I'm an arm cell, I'm a skin cell, I'm an eye cell. You can make it kind of like a brand new, fresh, whatever I'm going to be cell, which is essentially a stem cell, right? A stem cell is a cell before it defines itself. And then you can then tell it to try to make it to guide it become a brain cell.
That's exactly it. That's the concept. Yeah.
Oh, wow, that's freaking amazing. I can't even believe science can do that. That's pretty scary at the same time.
It is, but it's something that has been going on for a while. I mean, for at least 10 years.
Wow.
But I think what's the most amazing thing is that over these 10 years, we have been optimizing the entire process. And now not only can we create brain cells, we can create small structures that resemble the embryonic brain, and we call them brain organoids. Some people in the media, they call them mini brains. They do not look like a real brain. They are just like a small piece of a tissue, but contains some very specific cells that can create a network. So those neurons can communicate, and most importantly, they can reveal what goes wrong when someone has autism.
I see. So you take these stem cells, you guide them become brain cells, and then you can develop these little tiny mini brains based upon my skin. So you would have like a Felicia mini brain.
That's it. That's it. Because we capture your genome into those stem cells, the mini brains that we have in the lab is going to be Felicia's mini brains. Is specific to each individual.
Wow. I wish you could show us a mini brain, but we're not. I know you couldn't be in the lab because it's very busy there. But in the future, we'd like to see one of those mini brains. So
Absolutely.
you get these mini brains, and then your lab focuses on kind of examining those mini brains, and not only just with autism, but other types of research as well, right?
That is true. Yeah, we work on autism and other neurological conditions, some rare disorders, also epilepsy.
Okay.
And so what we do is we create those mini brains from a group of people who have autism or epilepsy, and then we develop them over time, and we compare to a control group most of the time. These are neurotypical, typically developing people who do not have the disease, and we ask what's different between those two groups. And for example, take autism. What we find is that the connections between the neurons inside the mini brain from someone with autism, these connections are different from the neurotypical group. These are the synapses where the neurons talk to each other. And most of the time in autism, we have a reduction in the number of synapses that are being formed. So that's a real, what we call a phenotype, or a distinction between the neurotypical.
Okay, so we have these brains that we can now compare to a basic type of everyday person, healthy human brain, and we're looking and comparing, and we're saying these are differences between the autistic brain and the typical brain. And that's, you're able to make those comparisons and then be able to understand a little bit more about how autism works in our brain. So how did you come to determine that CBD and autism was a worthwhile subject to study?
Yeah, because of our work on epilepsy, I interact with lots of families that have kids with seizures. And some of the families, they were telling me that CBD oil or hemp oil was good for them. They were reducing the number of seizures. Some families were claiming that they are out of seizures. This was, again, I mean, back in 2008, 2009. So I started paying attention to that. And then at the same time, kids with autism, the families were reporting that they were getting better. But, you know, I mean, the problem is this was all anecdotal. I mean, the families were telling me, and they were showing me the products, and the products were coming from everywhere. Some of them, I mean, they didn't even have a label on it. So the amount of CBD or THC in each one was quite variable. So I could not make any sense of that. So it was hard. I mean, the only thing that we had at the time was this empirical evidence that it might work for some people. So that's how I became interested on that, and I started using CBD in the lab and testing on these mini brains for seizure, for epilepsy. So now we know that for some specific types of seizures or epileptic conditions, CBD is very useful. And the only way to know that is to run what we call a clinical trial. So that's when you recruit subjects and test in a very controlled, organized, systematic way if the CBD or the drug will work or not for that specific condition. So for epilepsy, for seizures, we have those two conditions that are now approved. But for autism, no, we did not have. Was only recently where the NORDA Foundation and the Holistic Foundation gave a gift to UCSD to explore that. So we are running a clinical trial now, or we will start running a clinical trial this year on autism and CBD. And we are isolating autism from seizures to know if CBD can actually work for the symptoms of autism.
So seizures are a little bit different because like seizures, you can actually see a start and a stop, right? The seizure starts. If you give CBD, you can determine whether it stops based upon you giving the CBD. But with autism, it's different because there's a whole spectrum of conditions that are part of the autism, you know, prototype of person, right?
You are absolutely right. So autism spectrum, as the name suggests, is a spectrum. So you have people who are in the more severe side and people who are in the more less severe side. So I think the point here is to select a specific cohort. And by cohort, I mean the subjects who are going to be part of this research that would be as homogeneous as possible, as similar as possible among them. And we will characterize each one of them in a very precise way, in a very systematic way. And we're going to be looking for conditions such as repetitive behaviors, socialization, language, things that we can actually quantify. There is even measurements such as EEG or brain waves that we can record them before the treatment, so we can compare after the treatment if something changes. So a clinical trial like that is almost like in an individualized fashion. So you have to look for each subject and see if the drug is helping each one of them. So that's how we can make claims about symptoms of autism.
So that's why the mini brains are so important, because for every subject, there's going to be a mini brain that you can also make those comparisons. So I just wanted to talk a little bit about the subjects, right? So these are going to be children in this particular study, correct?
Yeah, we are recruiting about 30 boys, only boys this time. They will have 8 to 12 years old. And we are looking for the ones who have a very severe type of autism, the ones that have aggressive behavior, self-injuries, stereotypes, hyperactivity, anxiety, sleep disturbance, because these are the things that we can actually quantify.
Right. So you'll be able to see the difference. You'll really be able to see the difference with these subjects, because their conditions are so extreme. You'll be able to see a difference when you are doing the testing. So how does a study like this get put together? Like, what are the pieces of it? Like, you've got the children, and what are the other components?
Yeah. So there is a team of clinicians who's going to be recruiting those kids and performing all kind of characterization. For example, I mean, the EEGs we're going to be recording from them from time to time before even they start the treatment. There are other things that we can do, for example, to measure attention. We can use eye tracking devices to see if they can pay attention on a specific track. We can record their movement while they're doing like stereotype behavior. So all of that is going to be quantifiable and share with other researchers. So this is the clinician team. Then, I mean, a very interesting aspect of this study is, as you mentioned, we're going to be running an in vitro or in the lab clinical trial, because each one of them is going to have their own mini brains being reprogrammed and done in my lab. So we're going to follow how these mini brains evolved over time, and we're going to be treating these mini brains as well with CBD to see if we can see anything. Things that we can actually measure from the mini brains are these brain waves. We can see if the communication of these neurons are being affected by CBD, the number of synapses. These are parameters that would be impossible to look in vivo, because we cannot put a microscope inside each brain. So that's the value of have the parallel mini brain clinical trial going on. And finally, we have a third aspect of that, which is the Cannabis Research Center here at UCSD, who's going to be doing like an analysis of how much CBD is in their blood. This is what we call pharmacokinetics, how much they process that, and what would be the optimal dose. We're going to be controlling for all these parameters. Wow. So it's quite sophisticated studies, and really, to be honest, these are very expensive studies, and that's why, I mean, we need philanthropic donations to be able to perform that. Right. It's really hard for example, the NIH, our government agency, to support this kind of trial. We need support from outside.
Right. So this is a placebo-controlled, double-blind trial, which is the platinum what trials should be. This is the one that people would endeavor for. And these are the ones that could be used to validate information and to further research. And you bring up a very good point about the importance of the funding for this, because these types of trials can cost millions of dollars. And typically it's the pharmaceutical companies who fund these trials, right?
Yeah, yeah. Most of the time, I mean, it's the pharmaceutical companies who actually move on and perform the trial so they can sell the product as a FDA-approved drug. That's what happens with Epidiolex. This is the CBD version for seizures or epilepsy.
Right. So Epidiolex is actually a drug that's out right now. It's by GW Pharmaceuticals, I believe.
That's correct. Yeah.
And they have a drug that they basically patented that they're selling on the market. But this what work you're doing right now, this is really the early stages of seeing what could be done in regards to CBD and autism. I just wanted to reiterate, you're saying that there's the children, you have also the team that's creating the brains, your team, then you've got the team that's kind of testing the blood, the pharmacokinetics. That's a new word for me too. I'm like going to be like a PhD by the time we finish this. I just want you to coming down there. And all of these people together, these groups together are going to really provide all the different touch points that are going to make this valid information at the end of the day.
That's correct. Yeah.
Awesome. Okay. So I just really wanted to get in a little bit more about the mini brains because I'm so freaking infatuated with them. When you are talking with, when you have these subjects, they're going to be 30 children, and each child will have their own parallel universe of a mini brain that's an actual brain that will reflect who they are, their individual selves. You're going to be giving the children the CBD, and you're going to be giving the mini brains the CBD simultaneously.
Yeah. And as you can imagine, I can see, I mean, who is going to respond to the CBD and who's not going to respond much earlier than the clinical trial. The clinical trial might take like a year or so we can do the analysis, and everything was blind. But the analysis in my lab is going to be quite fast because those mini brains, they would be formed and ready to go in 6 months, and the treatment is quite fast as well. So what we expected for the clinical trial, it is that the CBD will probably not work in the same way for everyone. Maybe there are some people who respond really well, and you can see all the symptoms goes away or a reduction in the symptoms. For others, they might need more CBD, or the CBD will not even respond because they are not susceptible to CBD. So that's normal with any drug. But in the lab, I can actually adjust for each one of them, and I can learn why they are not responding to CBD. Is it a dose thing? So I can increase the dose of that specific subject to see if they start to respond. Or is that something on the genetic side? So I can sequence their genome and trying to figure out if they have all the right receptors, or if they have any other problems that would make them not responsive to CBD. So it starts to open more questions about how CBD works. So that's what we call a mechanistic way. So the clinical trial will go in parallel with the mechanism of CBD for each one of them. It's almost like a personalized medicine.
That's what I was going to say. It's personalized medicine. So you are going to say to Bobby, you know, this dose of CBD actually started to cause this effect. We went up to 100 milligrams or 200 milligrams, and the effect stopped. So this is your range, and these are the types of way that it's acting in your body. It's causing, you know, this part of your brain to react in this way, so that they'll actually be able to possibly take it and then use it more effectively at home.
That's correct. And obviously, I mean, because this is still very expensive, because it's all in academic side, they cannot create a mini brain for everybody in the world. But I think that's more or less the future of medicine. It is possible that in the future, not only for autism, but for other neurological conditions, so you're going to go to the doctor, and instead of prescribing right away a drug that might work, he's going to run a test on your mini brains in the lab, and then wait for the results. So he will know with more precision what is the dose, what is the medicine that will work for you.
Wow.
So I think this is the beginning of a big revolution in medicine.
That's so exciting, but scary at the same time, because you could just imagine all of the, the other issues of, well, is it a live brain? Who does it belong to? It's just, it gets a little bit out there, but that's like really, really powerful to be able to get that type of customized, personalized approach. So the CBD that you're using on the mini brains, is it going to be the same CBD you're using with the test subjects?
Yeah, so we're going to start with the synthetic CBD, and we're going to use exactly the same CBD in the mini brains and in the subjects. Obviously, the subjects will continue on that one, the same synthetic one throughout the clinical trial. We cannot change that. But in the mini brains, I can do whatever, because when I grow a mini brain from someone, it's not one mini brain, it's hundreds, if not thousands of them. So I can use each one to test different concentrations or different ratios between CBD and THC, for example. Maybe THC will work for some people. And I can just play around with experimental conditions to really understand better why that specific mini brain reacts or not to CBD.
So the synthetic CBD, how do you expect it's going to be different than a natural CBD? Do you expect already some variances in that?
To be honest, I don't think so. And we are just starting with the synthetic one. We could start with the natural one. So I don't think we're going to see major differences. There are some reports in the literature suggesting that in some specific conditions they might differ, but we don't know yet. I mean, I think the evidence is it's still immature. But what I think I can do is in the mini brains, I can try natural one, I can try the oil, I can try the extract. So it will inform us on the efficacy of the different concentrations of the different mixtures of different reagents from the hemp oil.
Right. So we'll be able to actually see just a totally separate benefit from your research, is to really kind of see whether, a synthetic CBD actually operates the same as a natural CBD, and then the full spectrum versus the isolate. So you're actually creating, this synthetic one is more of an isolate, correct?
That's correct. Yeah.
Okay. All right. So no full spectrum, no extra cannabinoids, just the CBD in an isolate.
Pure. Yeah.
Pure. Okay. So what parameters and tools are you using? So we talked about measuring the results. We have EKGs, we have EEGs, we have blood work. When you're getting this data, who's crunching the data? How are you, using the data after you get it?
Yeah. So most of the clinicians and psychologists will help us to put the data together. And then, as I mentioned, we're going to be sharing this data. So data that are very interesting to me is the EEG, the brain waves from the subject. So what I'm hoping to see is that after the treatment with CBD, their brain waves might change. And I want to see if this will recapitulate in the brain waves that I can record from our mini brains. Because these mini brains, I can also plate them on multi-electrode arrays, and these electrode arrays can capture the electrical activity. And I hope to compare them with the actual person. So there are many people involved. There are my collaborators who help us on translating the analysis of the cells in a dish to the brain of the people. These are people who are on cognitive neurosciences. They have been looking through EEGs for years, so they know exactly what to look for. So it's very collaborative. So we are all together on that. And then there are more biochemical readouts. For example, how long does it last, the exposure of CBD? And one thing is in the person, the other thing is in the mini brain, because in the mini brains, one of the limitations is you don't have a body. So the CBD is directly in touch with the brain cells. And on the subject, they will circulate into the entire body. So this is another thing that we'll have to take in account when we do this biochemical analysis.
Right. So those variances, because people would typically the subjects will take it under their tongue, right? And it'll absorb, some of it will absorb, but then some of it will go into their digestive system. So
Right.
that is the difference between the mini brain that's getting the CBD directly versus the other one that's being processed through the digestive system of the subjects. That's a variance.
Yeah, that's a variance. And we are fully aware of that. I mean, there's no other way or better way to do that. We'll just have to take that in consideration and be very honest during our analysis that these are the things that we cannot compare, or if we can compare, we have to make sure that these are the caveats doing this comparison.
There seem to be so many different variances and caveats. You know, how do you find that one line of this is true, this is clear, we can work with this information, and kind of put the other things to the side? It just seems to be that it could be overwhelming. I'm just thinking about doing the science project with my daughter this year, and it was much simpler, but there was still multiple variables there, and I was like, my head was kind of hurting. So this is a lot to deal with.
Yeah, and you know what? I mean, Felicia, at the end of the day, science is not done by a single lab. So there is all what we call replication. And things start to become a strong evidence when multiple sites, multiple labs start to see the same thing. So we build upon several evidence, and then something becomes like a scientific truth, which might never exist because all the conditions are variable. So I think we are contributing to the understanding, but there is no final truth, right? I mean, we will have to wait for other people to reproduce that in different conditions in other parts of the world. And that's why it's so important to have multi-site clinical trials going on at the same time.
So there are clinical trials happening at your location, as well as not the same trial, but different variations of trials. And you all will share information and overlap and see what works for other people's information, and then be able to publish it. So you, after this is done, you publish this to the journals.
Yeah, that's exactly what we do. I mean, because we are all from the University of California, so we are not tied to any pharmaceutical industry. This is all public available. So as soon as we are done with the studies, we publish, we do the analysis, and we publish all the raw data so people can check it out to make sure that what we did is correct. And the whole scientific community will have access to that. I'm expecting, because this is such a hot topic now, that many people would be looking at that data very carefully. And if there is any problem, anything that we might have missed, and remember, we are humans too, so we might make a mistake. So others will catch up and notify, and that's how science evolved.
Well, if we just created a mini brain of you, then it'll be perfect, and we won't have to worry about any mistakes. And then we'll just put a robot body on it, and we won't need you anymore. So at the end of the day, when everyday people are able to look at this public information, thanks to organizations like the NIH and the Holistic Research Foundation for funding these type of studies, they should be able to get what from it? They'll be able to see what dose you used, you know, possibly the type of CBD synthetic that you used, and maybe find that specific type somewhere. How can they use this and use this information for their own practical everyday use?
Well, I think the goals of the studies is first to show that CBD is safe and is effective in treating the behavior problems associated with autism, mainly severe autism. And I'm saying that this would be like our primary outcome. So we're looking for, as I mentioned to you, self-aggression, self-injury behaviors, repetitive behavior, things that the family would be able to tell, yeah, he did improve. So you don't need to be super smart to see that the kid improved. That's what we're looking for. And then, I mean, I would say that secondary outcomes from this research will tell us not only if it's safe, it's effective, but perhaps, I mean, what is the natural variability that we would expect it to see? How many people should respond? And hopefully we can tell why some are responding to the treatment and others don't. And more towards the basic science, exactly as you mentioned, we might be able to dose specific CBDs or even combinations of factors to personalize the treatment for each one of those subjects. going forward, once this is published, we hope again, I mean, if this is all become true and validated, that would be an effective treatment for autism. So the families will benefit by knowing that there is an FDA-approved medication that will help with severe types of autism. This is something that we have been missing for years. There is no current treatment for the behavioral symptoms of autism, apart from some drugs that has been repurposed from other diseases to treat autism, but nothing has been really designed to treat autism in the way that we're doing right now.
This is breakthrough because, like you said, people who have autism, the treatments now really are, hacked treatments from other drugs that, well, it works for this. We found that it can be effective in autism, and it probably comes with its own set of symptoms and additional counterindications as well. But this particular research will hopefully pave the way for additional research, maybe that will create a drug that will specifically help people who have autism.
That's correct. Yeah.
That is so exciting. I mean, you're at the ground. How does it feel to be at such a, ground floor? I mean, is this like where Nobel Prizes come from, where people are doing this type of great work? Because this is really important work for everyday people, and it's really groundbreaking.
Oh yeah, no, I feel it's a mix of excitement to be part of the team doing that, and also like very privileged, because this is a kind of information that it's only once upon a time that you have really this capability of translating everything that you do in the lab to actually help real people. Most of the translational research take several years. So we might be talking on something here that might go really fast into clinics.
Excellent. And we're expecting to hear about the results when? Like, is it another 10 months, 12 months?
We are starting to recruiting this year. The trial might last a year or so, and then probably another 6 months to analyze the data, writing a manuscript, making sure we cross all the T's, dot all the I's, and finally publish. Yeah, let's say like 2 years, 2 years and a half to have a final result.
Okay, so in the meantime, regular everyday people, children with autism, we know that CBD is, according to the World Health Organization, it's safe to be used up to very high doses, according to the World Health Organization. So people could feasibly do their own, study at home, right? It's safe to do, and they could titrate up and see how it's helping them.
Yeah, I think this is the good news. I mean, we're not dealing with something that is going to be really bad, but just to remind people that for some people, there are side effects. It's nothing as dramatic as we see with other drugs, which again, this is the good news. And I mean, doctors cannot prescribe CBD for autism, but there are CBD available out there. I mean, at least in some parts of the world where it's legal. And I think it's just a matter of looking for good ones that you have more indication that is working or not. Right. One of the things that we see for some families when they bring their product to us and we check, and in some of them we don't even see CBD at all. So
really, yeah. So you actually put these products in the lab and there's no CBD in them? That's
correct.
Wow, that is the problem right now we're having in the space. I mean, before your research comes out, hopefully there will be some regulation, because there is an overwhelming amount of brands. There is there's no consistency in terms of what's on the label, in terms of the dosage right. that are in the label, right? Or what's in the product. And you said that you've actually tested some that has nothing in the product.
That is true. Yeah.
Yeah.
I mean, we need better regulation. I mean, this is, I think right now is kind of a gray zone, and there is not, there is no fiscalization of those products. Nobody's like really testing them and validating them. So.
Mm-hmm. Well, this is wonderful. Wonderful. Kudos to you and your team. We are excited. And of course, we want to just tell everybody, consult a doctor. This is not medical advice, the whole caveat, blah, blah, blah. Talk to your doctor about CBD or, your health professional. In the meantime, Dr. Mutri, we appreciate you. Keep going forth. Don't be discouraged. You've got hard work to do, but we will be waiting on the other side when the results are in. And this is Cannaramic, and we hope we brought you cannabis like you've never seen it before. And continue to watch, because we're going to have more wonderful folks just like Dr. Mutri here on our upcoming speakers. Thank you so much, Doctor.